Hello, biologists. Congratulations on making it to lecture number 16. We're now going to begin talking about animals, and the first part of this is going to be all on the tree of life. This is a somewhat longer lecture, so I'm going to actually break this into two mini lectures, and I will post them both individually. So here we go. The study of animal diversity is an incredibly daunting task, and that's because there are so many animals on the planet. Fortunately, the terminology that we use to study the diversity of animals on this planet is quite universal. So let's go ahead and define a few terms before we begin. Taxonomy. We're going to use this word a lot. Taxonomy refers to the practice of classifying organisms. That is, coming up with compartments within which we can stick organisms that tend to look similar or have similarities about them. Then there's classification. This is grouping and categorizing all life on the planet. And this is how we come up with rules in order to compartmentalize things in a rational way. Then we have systematics. Systematics is a little different from the first two because it actually implies degrees of relatedness between species. And some species are more closely related than others, and we can figure this out based on physical features, those things that might be common about them. We also might use DNA. That's kind of a more recent development. And really, we've been doing this for a very long period of time. Aristotle is really the first guy to apply some real rigor to the study of animal and plant diversity, and he was doing this more than 2,000 years ago. And while we have gotten a little bit more sophisticated nowadays, we really do use a very similar way of doing things to that of Aristotle. For example, he came up with names that he called genera, and Felis is an example of a genera that Aristotle came up with. Felis referred to all cats. He looked out, saw all cats, recognized that there was something similar about them, and figured they must fit nice and neatly into a compartment of their own. Same thing with this over here, equus. Horses and those things that were horse-like, he kind of put them all into a category. But it wasn't until until about 1700 years later, when Carolus Linnaeus comes along, also known as Carl Linnaeus, and he invents this system that we know as binomial naming. Binomial naming is a way of ascribing a scientific name to a unique species, one that will never change and that is recognized all across the world. And he chose to do this naming system in the Latin language. Why would he choose Latin? Who speaks Latin nowadays? Other than the Pope and a few other people, very few people actually speak Latin. It's considered a dead language. So that's really nice because when you name an organism in a dead language, it means that language or the name will never change in its meaning. That is the reason why scientific names for species are in Latin. Each species has a unique two-part name. Scientific names can be hard to pronounce sometimes because they are in Latin, but they usually have some sort of meaning to them. So for example, here's one, Paralabrax maculatofasciatus. Okay, you don't need to memorize that, but that is the scientific name for the spotted bay bass. A couple of things about this name I want you to recognize. There are two names to them, Paralabrax, and then the second name is Maculatofasciatus. Notice that the genus name, which is the first name here, has a capital at the beginning of the word. The second name is the species name, and it is not capitalized. This is the spotted bay bass. Now, why don't we just call it the spotted bay bass? Well, because I'm sure that somewhere in the world there are at least a dozen other fish that have the exact same name, spotted bay bass. And while there might be a gazillion fish out there that have the same common name, spotted bay bass, there's only one fish in the entire world, or one species in the world, that is named Paralabrax maculatofasciatus. And this is what it looks like, you guys. That is Paralabrax maculatofasciatus, and that is yours truly holding him in the back of San Diego Bay before I let him go. There are multiple classification levels that are essentially nested one within another. Each one of these is referred to as a taxon. If you're referring to more than one classification level, we call it a taxa, T-A-X-A. That's the plural of taxon. And this really helps us out. For example, take a look at this picture here. What if I said, hey, look at that. It's a purplish, brown, bumpy, inanimate star-like thing. Okay, that's not going to help either one of us figure out what this organism actually is. That might be a descriptive way to describe it, but it's not going to help us identify it. So instead, we would say, hey, look at that. It's Pisaster ocratius. That is the genus and species name for this particular sea star. It is much more specific, and we have actual characteristics that define Pisaster ocratius. It really helps out by giving scientific names to things, even though they're kind of a mouthful oftentimes. Scientific names help us avoid confusion, the confusion that would occur if you were only using common names. If I was speaking to my colleague in China and I was saying, oh yeah, I'm studying a red fish, okay, well they've seen redfish too, haven't they? They've probably seen dozens of redfish, as have I. So how do we know we're talking about the same thing? If we say Pisaster ocratius, then we know exactly which sea star we're talking about. Now here is the classification scheme, that nested classification scheme that I was mentioning. And you are going to have to reproduce this on an exam. It goes like this: kingdom, phylum, class, order, family, genus, species. Kingdom is the most generic of all of the classifications. Kingdom refers to a very broad group of organisms. Phylum is a little bit more specific than kingdom, but it's still considered a pretty broad distinction. At the very bottom here, you have species. Species is the most specific taxonomic identifier that we have. And when we are referring to a scientific name given to an organism, we are actually referring to its genus and species, these two things down here. Kind of like using your first and last name, but in reverse. In this case, genus would refer to something akin to your last name. Think about your last name. Well, there's more than one person with your last name, but not very many, right? And by adding your species name, well, that makes it very specific, right? By adding your first name, let's say, then that makes it very specific. So let's go ahead and show an example of this. There you go, that's us. We are Homo sapiens. Homo sapiens is our genus and species, which we simply refer to as our scientific name. The family within which we belong is the Hominidae. Hominidae includes other genus names and hence other species names. None that is alive today, but throughout the human history, we actually have had other genera that have fallen within this same family. The Hominidae fall underneath the primates, and there are certainly other families alive today that are also nested within the primates. All primates, and actually a lot of other animals, would fall under the heading of mammals. Mammals include primates, they include rats, they include dolphins, and lots of other things. Mammals are also one subset of the phylum Chordata, and Chordata is nested within the kingdom Animalia. Now, how are you ever going to remember this order here? I mean, that's a mouthful. Kingdom, phylum, class, order, family, genus, species. Well, we like to use a mnemonic. And a mnemonic is kind of a way to jog your memory to remember the proper order. And this is it up here. Kings play chess on fine green silk. Kings play chess on fine green silk. If you can say that to yourself 100 times over, you'll get it right. The first letter of each of these words actually refers to the first letter of the classification scheme that I have over here. So, kingdom, phylum, class, order, family, genus, species. And then we have our human here. The common name is simply the name that is colloquially used to describe the species. And there is a Homo sapiens. That's actually our governator before he was the governator. And if we were to now compare this to another organism which shares some degree of relatedness with us but is not the same as we are, maybe we could choose this guy. Here you go. This is a killer whale, or Orchinus orca. Orchinus orca is the genus and species, or the scientific name. Notice that the genus is capitalized, but the species name is never capitalized. Now, we are the same as this guy in that we are in the same kingdom, we are also in the same phylum, and we're in the same class. However, we belong to a different order. We belong to the primates, they belong to the cetacea. And by this methodology, you can actually compare yourself to just about any other organism on the planet. Here we have the California spiny lobster, Panulirus interruptus. And its taxonomy is such that we are only similar to it in its kingdom level. There. Let's go ahead and take a look at some examples here. How about fish? We already talked about one member of this genus, Paralabrax, so let me go ahead and compare it with a couple of other species that we find here in Southern California. This is the one that we saw before, spotted sand bass, Paralabrax maculatofasciatus, and it is very similar to Paralabrax clathratus. Same genus, different species. Paralabrax nebulifer, also same genus, different species. And in this way, we can pretty much identify just about any animal on the planet. Have we named all the animals on the planet? Not even close. We have a lot more to go, but at least among the ones that we know, we are pretty confident on the placement of all of these taxonomic names. This is Carcharodon carcharias, the great white shark, and this is Canis lupus. Okay, not the human, that would be me, but the dog. That's my dog, and she belongs to Canis lupus. That's the genus and species. Now, it turns out that sometimes we have to invent new categories, because in this case, the genus and species isn't enough to identify this organism as being different from, let's say, a wolf, which it turns out is very similar. So we have to come up with something called a subspecies, which fits underneath the species, and it gives it a third name, familiaris. So Canis lupus familiaris is the full scientific name for the domesticated dog. And in this way, we have been able to essentially create a family tree, also using DNA and RNA analysis. And in this particular case, the tree that you're looking at was made with rRNA, that is ribosomal RNA. And if you remember ribosomal RNA in your readings, you'll remember that RNA cannot change very much. tRNA and rRNA cannot change very much, otherwise it totally ruins the function of rRNA and tRNA. So it tends to persist over evolutionary time, and it remains the same. This makes it a really good tool for trying to create family trees. And this is really kind of interesting. It means that all organisms on the planet can be easily divided up into three major groups: the bacteria, the archaea, and the eukarya. We belong to the eukarya. Remember that? We are the eukaryotes. We have the more complex cell. Prokaryotes fall into these two groups, the bacteria and the archaea. And I think the thing that is most humbling here is that the bacteria and the archaea tree are huge, and the eukarya tree is only is actually dwarfed by the two of these together. And what's even more humbling is that among all eukaryotes, animals are just one little sprig on the tree of life. And with that, and that's you right there. With that now, we are going to go directly into the invertebrates. That's right. We are now starting to discuss animals, and the first animal groups are going to be the simplest of the animal groups. So let me go ahead and start with an outline to give you an idea of what it is that we're going to be covering here. We're going to talk about evolutionary trends within the animals, and we're going to use those evolutionary trends to help us define one group of animals compared to another. Now, the differences from one major group of animals to another is something that I expect you to know for an exam. So the first thing we're going to cover is the emergence of multicellularity, that is going from being a single-celled organism to being multicelled. We're going to talk about body plan traits, the symmetry of an organism. We're going to talk about tissue levels of organization, the presence or absence of body cavities, and then we're going to deal with unique features about each animal group that will help you identify them. Then we're going to go over the major invertebrate phyla. We're going to be skipping over some of the minor ones, the ones that you probably will never encounter in your life, and stick only with the major ones. So here we go. Once upon a time, everything on the planet was unicellular, was made up of a single cell. And, you know, if it were possible to be a bigger cell than the smaller cells, well, maybe that's a good thing, because then you could swim around as a single cell and eat those cells that are smaller than you. So here we have a nice little cell, and along comes the Pac-Man cell and eats him. Well, that's because he was a much bigger cell. This little Pac-Man cell is much bigger, kind of the big bully in the primordial soup. And, well, that's kind of frustrating if you're the small cell. Now, fortunately, you are going to get together with your buddies here, and you're going to kind of interact with them in a way that, that benefits you all. And in this way, that Pac-Man cell is no longer the predator of you, but you guys collectively can work together to eliminate the Pac-Man cell. Well, okay, this is just kind of a very simple, cartoony way of illustrating the advantage of being multicellular. It is possible that over evolutionary time, about 3 billion years ago, the first cells began to cooperate with each other. They weren't truly multicellular for a while, but they at least began to cooperate. And in so doing, organisms began to share genetic information and eventually become multicellular. So evolving from being unicellular to being multicellular is one evolutionary trend that begins in the animal kingdom. And so animals all evolved from single-celled protists more than 600 million years ago, and we refer to animals or multicellular animals as being metazoa. Metazoa means multicellular animals. And the diversity of the metazoa, the diversity of the members of the animal kingdom, is huge. Now, one distinction here is that all animals are heterotrophs. Now, if you remember from the photosynthesis lecture, heterotrophs are different from autotrophs. We talked about photoautotrophs as being those animals that can photosynthesize. Well, animals can't do that. We must get our energy and our nutrients from eating other things around us, whether that's eating vegetables or eating animals. And so we refer to ourselves as heterotrophs. Most kinds of different animal forms are actually found in the ocean. So the ocean is a very important place for animal diversity. 97% of all animals on the planet lack a backbone, and the term for that is invertebrate. So invertebrate animals are actually far more common than vertebrate animals. This here is kind of a very general family tree of all animal life on the planet. And you'll recognize a few animals here that you've probably seen, including clams, maybe anemones, crabs, but there's probably a lot of things on here that you don't recognize. We're not going to be covering all of these animals. We're only going to be covering about half of them. But I want to draw your attention over to here, the chordata. This is where you are. You're on this side of the scale, and this is my little vertebrate. Turns out that vertebrates fall within the category chordata. Now, what traits do we use to classify animals? When we go through each of these animal groups, I want you to ask yourself these questions and really refer to each one of these with every individual animal group. Number one, symmetry. What kind of symmetry does this new group of animals have? And we'll describe what symmetry is. Number two, how many tissue layers does this animal have? Number three, does this animal possess a body cavity? And if so, what kind of body cavity? And finally, number 4, what are the unique characteristics about this animal that make it unique from any other group of animals on the planet? Okay, so let's come to that first one there, symmetry. Symmetry really helps us define the basic body form of an organism. And all animals on the planet, we can really divide them up, for the most part, into 3 major groups. The first group is being asymmetrical. Anytime you see the letter A in front of it, it means not having it or not possessing it. So an asymmetrical animal is an animal that doesn't possess symmetry. That is, it doesn't possess a recognizable shape or order to the physical orientation of the body. Number 2, radial symmetry. Radial symmetry are organisms like this anemone right here that possess a top and a bottom but don't have a clear front or back. Finally, there's bilateral symmetry. Bilateral symmetry are organisms kind of like you and me. They have a front, a back, top, left, and the right and right and left halves of the organism are mirror images. You think about it, the left side of your body and the right side of your body are kind of mirror images of each other. This worm is kind of the same way. This is an example of a bilateral organism. Now, the second distinctor here is tissue layers. Turns out that different animals have different levels of tissue organization. And this stems all the way back from the organism's embryonic development, all the way from back when it's a fertilized cell and multiplying as multiple cells until it becomes a fetus, and then a fetus developing into a baby, and the baby developing into an adult. All of the organ systems that you have in your body, your skin, your liver, your bones, your muscles, it is all derived from tissues. And so tissues really are just specialized cells that all kind of function together for a similar use. Some animals have no tissue level of organization whatsoever. I know that sounds kind of strange, but there's actually a group of animals that are bona fide animals that do not possess true tissues. Some animals, all of their organ systems are derived from only 2 tissue layers. And since it's 2 tissue layers, the term that we use is diploblastic. Diplo means 2, blastic refers to tissues. Still others, including us, use 3 or have 3 tissue layers from which all of your organ systems are derived during your embryonic development. Triplo means 3, again, blastic means tissue layers. So certain organisms are triploblastic, others are diploblastic, and still one group of organisms, it has no tissue level of organization at all. Now, the third distinctor, body cavities. Does this organism have a body cavity? Well, during development, organisms can actually form what we can see to be a fluid-filled cavity. And this fluid-filled cavity is the place within which your internal organs kind of are all floating. Now, the best analogy here is to think of a donut. A donut has a hole that goes through the middle, right? That's kind of like your gut. And you might ask yourself, well, what is the outside of the donut? The hole that goes through the middle, is that outside? Well, technically, yes it is. It's the outside because if you picture your donut, the inside hole is still touching the air. If you want to get inside the donut, you have to actually break the donut apart, and all of that yummy goodness on the inside, all of the doughy, bready part of the donut, that's internal. You are built just like that. Your gut is technically outside. Here's your gut. It's running through your body. You are essentially a tube within a tube. That's how you're constructed. So certain organisms don't have this cavity between their gut and their body wall. Others have a very distinctive cavity within which their gut kind of extends down through the middle. Let me show you kind of how this works. I have this hollow tube here, kind of as an example to keep that in mind. Everything that is metal, that is actually on the inside of the metal here, would be considered internal. But the gut, which would be the tube, the hollow tube running down the middle, that is still kind of technically external. There's three different categories of body cavities. The first one is being acoelomate. A, again, not having, and coelom refers to the body cavity. So an acoelomate organism is one that does not possess a true body cavity. The body cavity is totally absent. And it means that the space that is between the tube within a tube, that is the outer tube that makes up the body, and the inner tube, which is your gut, is really just solid connective tissue. And I'm going to give you an example of one here in a little bit. Pseudocoelomate organisms are organisms that do possess a somewhat of a coelom. They do possess somewhat of a body cavity, but it lacks an individual lining known as a mesoderm lining. And I'm going to talk about the importance of mesoderm here when I refer to this next group, a true coelomate or a coelomate organism. These are organisms that do have a true body cavity that is lined with mesoderm. And the nice thing about mesoderm is actually the cellophane wrapper within which your organs actually sit. If you were actually doing surgery on somebody and you were opening up their abdomen, you'd take your scalpel and you'd cut open the skin, and what would you see under there? You'd see muscle. Then you cut open the muscle and you open that up. What are you looking at? The organs? No, you're not looking at the organs yet. Instead, what you're looking at is a cellophane container within which the organs are all laying. You have to open that cellophane container to get into the organs. This is known as mesoderm, or also peritoneum. Peritoneum is the human term that we use. Mesoderm is the embryonic name for that peritoneum. And so the nice thing about having all your organs wrapped in individual wrappers is that all of the physiology that is occurring in all of those organs can be maintained separately that way. That makes it very, very convenient, and organisms that have a true coelom are usually quite active, and actually the more derived organisms, those organisms that have evolved a little bit later. So again, let's take a look. Here we have an acoelomate, a flatworm. Flatworms have, here's their gut. Between their gut and their outer wall, this whole area here is just loaded with connective tissue. It's not an actual cavity. A pseudocoelomate, on the other hand, here is the gut running down through the middle of the body, and this is a fluid-filled cavity on the outside of that. That's a pseudocoelom. This is a true coelom. Here's the body cavity again, but all of the organs are essentially wrapped in their own mesoderm lining, again, also known as peritoneum. Now, let's move on to the first group of invertebrates, the phylum Porifera. So notice here that we're talking about the phylum level here. We're talking about all animals that fall within this phylum, Porifera. Porifera refers to sponges. These are all marine organisms that you're looking at in these pictures, although there are a few freshwater sponges as well. But sponges are pretty much all aquatic, either marine or freshwater. And of course the most famous sponge of all, this is SpongeBob right here. And poriferans are really interesting creatures. Pori refers to holes. Ifera means to hold or to have. So these are organisms that have multiple microscopic holes that allow water to flow in through their body. The phylum Porifera has some really interesting characteristics about it, so what we're going to do is go through all those characteristics that I mentioned in the last few slides and revisit each one of them for the phylum Porifera. This is the kind of stuff you are going to need to know for the next midterm. Let's define some characteristics. Number one, we do know that it's multicellular. That's what makes it an animal. But what about its symmetry? A sponge does not really have a defined symmetry. How about cellular specialization? Well, it does have cellular specialization, but it doesn't have true tissues. It does not have an organ level of construction. So this is an entire group of organisms that does not possess, it doesn't possess true tissues. Now, what about specializations in this group? Well, the one thing that is unique about poriferans that we don't see in any other group is that these guys feed with these cells that are filter feeding cells known as choanocytes. Choanocytes. And choano refers to collar, as in like the collar of a button-up shirt. And cyte means cell. So choanocytes are collar cells. And I'm going to show you a picture of a collar cell. One other key feature of this group that is kind of unique to them is the structural material that they are made of. They're made of a protein known as spongin and a bunch of hard calcium-rich or also silicate-rich materials known as spicules. Spicules look like sharp little needles, but they're microscopic and they make a gazillion of them. Mixed together with the spongin, you end up with a sponge body. This is the sponge's morphology. This is kind of what it looks like. It has a large hole at the top of each one of these little bodies, and the water flows through these small holes here in the ostium. Ostium referred to the small pores along the side, and the water's flowing in through all these little ostia, and then it flows out through that large hole at the top, the osculum. It's kind of like this. The water flows in, and it flows out, kind of like that. And again, the unique feature here, the thing that kind of defines this group, is the presence of this choanocyte or collar cell. They line the inside of the sponge and they beat this flagellum right here, this whip-like structure right here, beats and beats and beats, and it sets up a water current which it draws the water into the sponge. And in so doing, little particles and any other things that the sponge might like to eat get stuck on the collar here of the cells. And this is how the organism eats. It's actually a filter feeder. Now the structural materials, again, of the sponge consist of spongin. This is what spongin looks like when you dissolve all of the spines away, all the spicules away. And this is what the spicules look like when you dissolve away all of the spongin. And spicules, again, look kind of sharp, and they are usually made out of calcium carbonate, but sometimes they can even be made out of silicate. Silicate is glass, which is pretty amazing that there's an animal that is able to secrete glass. Now, let's move from the porifera to our next group of invertebrates, the cnidarians. Phylum Cnidaria. This is our second group of organisms, and we are now going to do the same exercise as what we did with the sponges. Oh, and by the way, I should say, I have put videos up on the course website for each one of these major phyla. So make sure that you go and check out those videos. They should be quite fun for you. Let's do the same exercise, though, with Phylum Cnidaria. We'll go through the defining characteristics. Number one, what kind of symmetry do they have? Cnidarians have radial symmetry. That is, they are built like a pizza. You can cut them down the middle any way, and one side will end up looking like the other. So they're round. They are radially symmetrical. And unlike the poriferans, cnidarians actually do have a tissue level of organization. That is, that these guys actually are derived from two tissue layers. They are diploblastic. And we have a name for the inside and the outside tissue layers. The ectoderm, ecto means outside, derm refers to skin, so this is ectoderm, and inside is known as either the endoderm or the gastroderm. Either term is actually correct. Gastroderm implies that the inner cell lining is used for the purpose of eating or absorbing food. Sandwiched between this ectoderm and the gastroderm is a non-cellular or acellular gelatinous material known as mesoglia. So mesoglia is what gives this jellyfish here its name. It's what makes it seem gelatinous. These guys do not have a full gut. They don't have a mouth and an anus like you do. They have only a mouth. So the food goes in, and then any waste product has to go out the same way it came in. So we just call that a blind gut. Now the one feature about this group, about the cnidarians, that is unique, no other group of organisms has it, is the presence of nematocysts. Nematocysts also are what make cnidarians famous because they are the stinging cells. They are what cause jellyfish to sting you, or anemones. Anemones can sting you too. And these stinging cells are used for feeding or for defense. And the stinging cells look kind of like this. This right here is a Portuguese man of war. You may have heard of this kind of cnidarian. It floats kind of at the sea surface with all of its tentacles hanging down, and they catch fish and also other small organisms, and they eat them. But they are immobilizing them with these stinging cells, these nematocysts. And the nematocysts are essentially a hollow cell that's under pressure that has an inverted harpoon in it. That harpoon is hollow, and if it injects into you, then it's basically releasing toxins into you that cause your bodies to scream in pain. It really, really hurts. Here's the trigger, and when you tap that trigger, the whole top of this thing pops off, and out comes protruding this venomous stinger. I've been stung by them many times. It's not very fun, so you should be very careful about around a jellyfish that you might find washed up on the beach. Now, there are 4 major classes within the phylum Cnidarian. Again, I want to draw you back to King's Play Chess on Fine Green Silk. Kingdom, phylum, class, order, family, genus, species. We're at the phylum level here talking about cnidarians. And within the phylum Cnidarian, there are 4 classes of cnidarians. The first one are the hydrozoa. The hydrozoa refer to a group of anemone-like organisms that live on the bottom, but also can produce little swimming jellyfish. And also the Portuguese man of war, which is known as a siphonophore, is also found in this group. The second group is anthozoa. Anthozoans refer to the larger anemones, like what you might find here at the tide pools at our local rocky beaches, and also corals. Corals fall within the anthozoa as well. Then there's the jellyfish proper. These large, charismatic jellyfish that swim in the ocean fall in the class Scyphozoa. And finally, there's one group of jellies known as the box jellies, which fall into the class Cubozoa. Box jellies are the ones that are most venomous out of all of them and actually kill a small number of people every single year. So let me show you the diversity of members of these four classes. Here's the first one, Hydrozoa. And the freshwater hydra that you see here is one that we often see in lab, and but there's a lot of marine examples of hydrozoa as well, ones that float on the sea surface, and also ones that swim around. So it means that all 3 varieties here of cnidarians are found within the hydrozoa. Oddly enough, this one up here, that's not one organism. That's actually multiple organisms working together in a huge colony. So each one of these little pieces here is an individual organism, and the float that you see at the top is another animal that has been modified to be a float. So in hydrozoa, you see all 3 types. You see the colonies, you see individual polyps. Polyps are the ones that kind of attach themselves to the bottom, and then a medusa is the kind that actually swims up in the water column. Medusa is reminiscent of the Greek myth, where the Medusa demigod essentially has the snakes for hair, so it looks kind of like that. Now the hydrozoan life cycle is actually quite interesting. Many hydrozoans, not all of them, but many of them exhibit both a polyp stage and then also a swimming medusa stage. These polyps right over here produce a little swimming stage, which eventually grows up to be an adult medusa. These medusa here are male and female separate, one producing sperm, the other producing eggs. These guys are tiny too, by the way. Little tiny jellies swimming around. You almost can't see them with your eyes. But they're producing sperm and eggs, which gets together and fertilizes an egg, which eventually divides up to become a planula larvae. And this planula larvae then comes down, settles on the ground, and grows up to be a new polyp. And this new polyp eventually differentiates and becomes a colony of polyps. So this is the typical life cycle of a hydrozoan, going from being a polyp organism to being a medusa organism back to being a polyp organism. Class Scyphozoa is the third class within this group, Cnidaria, and these are the big charismatic jellies that swim around right off of our coast. Then there's the Anthozoa. Anthozoa was the second group that I actually mentioned in that list, and these include the corals. Here's a coral, and here's an anemone over here, and you'll notice the color of these guys. This one's green, and this one's kind of a tannish brown yellow color, and that's because these guys actually have a symbiotic relationship with an algae. There's an algae that actually lives in their tissue and photosynthesizes. So while animals are all heterotrophs, that is, they don't photosynthesize, many of them do have relationships with algae which do photosynthesize and release the byproducts of photosynthesis directly into the cells of the coral, which is kind of a nice deal. Now the last group is this class Cubozoa. And what I'm showing you here is one of the nastiest cubozoans. This one has actually caught a krill, and it's eating a krill right here. Krill look very similar to shrimp, so it's got a little shrimp here that it's eating. And cubozoans are just nasty little beasts that are just amazing swimming around. This is Chironomyx fleckeri. You don't need to memorize that, but that's the genus and species name for this particular cubozoan. And it actually kills about 5 people every year, mostly in Australia, and most of the people that it kills tend to be small children or older adults, basically people who lack the immune system or the body to be able to handle a major sting like this. This is Tennille. Tennille's a 5-year-old little girl who got really wrapped up with one of these things, and she managed to not die, which is a good thing. But it can certainly do some damage to you. Class Cubozoa. Next group. We're now going to cover those organisms here, over here, that possess 3 tissue layers. All of the organisms up until now have either been no tissue layers, as in the poriferans, or only 2 tissue layers, as in the cnidarians. Now, from here on out, all of the organisms we're going to deal with have 3 tissue layers. So this is a really good dividing line here for evolutionary development. And the first group we're going to talk about here is the phylum Platyhelminthes, the flatworms. We can ask all the same questions that we have for the previous 2 phyla. What is their symmetry? Well, Platyhelminthes are the first group of bilateral organisms. By the way, let me go ahead and break down the name here. Platy means flat, and helminth refers to worms. So these are the flatworms. So they are bilaterally symmetrical. As we mentioned before, they are triploblastic. They are the first organisms to be based on 3 tissue levels of organization. However, they are still acoelomates. They do not possess a true body coelom. They also do not possess a complete gut. They have this incomplete gut. However, they do have cephalization. Cephala refers to head. So cephalization means the formation of a head. This is one of the advantages of being bilateral. If you are bilateral, it means invariably you're going to have a head end and a tail end and a left and a right and a top and a bottom. Having a head end, having cephalization is actually an important development for organisms that move around. When you're moving around, it means that all your sensory structures are actually encountering the world first. Think about it. Where are all your sensory structures? What do you think? You have 5 senses, right? You have smell, taste, sight, hearing, and touch. And 4 of those are concentrated on your head. I mean, technically you can even touch with your head if you wanted to, so you could do all 5 things with your head, but the fact that they're all there tells you something. Your head, or the head of all organisms, tends to be the part of the body that encounters the world first, and so keeps you out of danger. There are 3 major classes within the Platyhelminthes, and we're going to cover all 3 of them just very, very briefly. The first one are turbellarians. These are free-living flatworms like the one you're looking at here on the right, and they glide around in marine environments and in freshwater environments, and even a few are found on land. The last two groups here, the Trematode and the Cestoda, are both parasitic. They include the tapeworms, the bottom one here, and the flukes, the trematodes. And flatworms undergo some pretty interesting behaviors. Very, very interesting behaviors. You're looking at two flatworms interacting here. And you might say to yourself, well, what's going on here? Well, obviously they're dancing around, right? Okay, maybe not dancing. Maybe I wouldn't put it that way. They're actually getting ready to mate. And this is actually an interesting development within the platyhelminthes, within the free-living turbellarian platyhelminthes, is that these guys are hermaphroditic. That is, they possess both male and female parts. Now let me ask you a question. If you are a male, how much energy do you put into the development of an embryo? Well, actually, the answer, not very much. I mean, you drop off your sperm, and really you just sit and you wait for all the magic to happen in the female, right? If you are the female, then you have to pour huge amounts of energy into the developing embryo inside of you. Well, if you're a hermaphrodite, which would you rather be, the male or the female? From an evolutionary perspective, you'd prefer to be the male because you can mate and just make some new sperm, and within about 15-20 minutes you could be off mating with another one. Being the female means that you can now not mate for the next 3 weeks, and you have to develop your eggs inside your body and take care of them and put all that energy into it. So what these guys are doing here is duking it out with their penises. They're doing what is known as penis fencing. And what they're essentially doing is squaring off and trying to jab into the other individual to inject its sperm without getting jabbed by their opponent. And the one who loses ends up having to put all that energy into developing eggs, whereas the winner gets to crawl off and mate again. Go check out the penis fencing video that I have included on the course website. Class Trematode, this is that second group of flatworms. They are all endoparasites. That is, they live inside of their hosts. Many trematodes actually have multi-host life cycles. That is, their life cycle can only be completed if they have all of the hosts involved. I'm going to show you an example of that. Here you have a trematode by the name of Euhaplorchus californiensis. Don't memorize that. You don't have to. But it lives in the estuaries here in Southern California. The adult worms look like this, and the adult worms actually live in the gut of this poor bird. It's actually not a big deal. It doesn't really do too much damage to the bird. In fact, most often the bird doesn't even know it has those adult trematodes living in its stomach. The trematodes get together, they mate, they lay eggs, and the eggs end up passing out with the feces of the bird. When the eggs hit the water here, they hatch, releasing a swimming stage known as a miracidium. And this miracidium swimming stage seeks out the next intermediate host. In this case, it's the California horned snail, right here. They burrow into this snail's foot, and they begin to asexually divide. They turn into two parasites. Two parasites turn into four parasites. Four turns into 8. 8 becomes 16. 32, etc., until this snail here is loaded with parasites. Eventually what happens is the parasites leave in the form of a swimming stage known as a cercaria. It actually has a tail. Many cercaria actually have eyes, like what you're looking at here, so that they can see their next host. The cercaria then seek out the next intermediate host, which is the California killifish. This little fish lives in our estuaries, swimming around, and it encounters a cercaria without even really knowing it, and the cercaria burrows into its gills, travels through its blood system, and eventually creates a cyst inside of its body. And the life cycle is completed when the bird comes along, eats the fish, releasing this waiting stage here called a metacercaria, and the metacercaria hatches, and the worms eventually grow up to be an adult again. Now, the interesting part to this story is that you have a swimming stage, which leaves the bird, the egg hatches, and this swimming stage seeks out this host. Then you have another swimming stage, which leaves the snail seeking out the fish. What you don't have is a swimming stage that leaves the fish. So, really, this parasite requires this predator-prey interaction. It requires this bird to come along and eat this fish. So it makes sense that anything that that parasite can do to enhance the chance that this fish is going to get eaten by this bird, well, that's going to be conserved over evolutionary time, right? Well, that's precisely what happens. It turns out that this parasite insists on the brain of this fish, changing its behavior and rendering it more susceptible to being eaten by this bird. Let me show you. Here's that killifish. If you cut away the top of its head, voila, you're looking at the brain. Okay, actually, you're not looking at the brain. You can't see the brain because it's under a layer of cysts, of parasite cysts. There are about 5,000 cysts on this poor fish's brain right here. Actually, the brain, if you were looking at the brain, it would be this white color like over here. This is the spinal column, and it's coming in, and the brain is completely covered by parasites. Pretty potent way to affect behavior, don't you think? Yeah, I think so too. Okay, we're going to now stop here, and I'm going to pick it up with a second lecture. We're going to continue on with the next phylum. That would be the phylum Mollusca in the next lecture. So I'll see you all in cyberspace.